And I'm really delighted that this Lovestone agreed to give the second research task tonight, and his best role in psychiatry. And his title is an interesting title. It's a novel about shaggy, a translational journey in Alzheimer's disease research. So very much like to welcome Professor Lovestone as guest lecturer. Thanks so much.
Thank you very much. Just between you and me, I never intended to be an old age psychiatrist. In fact, I've got a terrible memory of when I was a medical student, an unimaginable time ago. And I was in I guess I would have been in my final year, my fifth year, and I was in some hospital. I really wanted to, I don't know, be a surgeon or a cardiologist. Actually, what I really wanted to be was an immunologist. And I was on some ward or lunch break, and I'm queuing up in the canteen, and I was having that medical student conversation that you're probably beginning to have. So the person in front of me was a guy in front of me in the queue, was in the year above me, so he was doing his house check. Oh, the doctor. So I was having a chat to this guy, and I said, so what are you going to do? And what are you going to be when you grow up? When you really qualify and you've done your house jobs, like Y1 stuff, what are you going to do as a career? And he said to me, I'm going to be an old age psychiatrist. What? Loser? I couldn't imagine anything worse that you could be, and here I am. So I honestly don't know how it happened. And I think it's probably true to say I've never made a career choice or a career decision at any point. I've drifted from one thing to another, and I've kind of followed the interest. So I've sort of done at every stage what seemed to me to be the most interesting thing to do. And I've never yet regretted anything, but it's also true to say I've never planned a thing. So I thought what I'd do is take an opportunity to tell you a little bit about the research that I've ended up doing. And this is a research that goes back now, I know exactly when it started. It started 23 and a half years ago. And I remember it was 23 and a half years ago because that was the time my first daughter was born. So, and I just happened to remember she was born and I started my research career pretty much at the same time. So when she was a toddler, or 2 or 3 years old, she used to come into the lab in the evenings when I was working on the cells, some of the experiments I'm going to show you. She, like you, is now a medical student. So a quarter of a century later, it's funny how these things come around. And one of the reasons why I wanted to work, why it seems to me to be a sensible thing to work on, Alzheimer's disease, is just a fantastically costly disorder. The person who bears the biggest brunt of that cost is, of course, the patient, who loses their memory, loses other cognitive abilities, and eventually loses their ability to look after themselves. And that's the biggest cost. The next biggest cost is, of course, to the family. And they lose, to a certain extent, they lose the person that they've loved all their lives. They lose a lot of time and effort that has to be put into it. And they lose a lot, and I, like many of you, have had the experience of having a close relative with Alzheimer's disease. Long after I started work on this condition, that wasn't at all why I chose to do it. But I saw at very close hand the costs to the family there. But there's also an economic cost. I'm not going to read these things out for you, but the economic cost is absolutely huge. If you turned the amount of money the world spends on Alzheimer's disease into a company, then Alzheimer's disease PLC would have twice the turnover of ExxonMobil, one of the biggest companies in the world. If you turn the money the world spends on Alzheimer's disease into an economy, then the economy of Alzheimer's disease would be bigger than that of Indonesia. Absolutely huge. And it is set to rise, if not exponentially, very rapidly indeed. And it's not going to be us that bears the brunt of that, because we already live in a very old society. If you look at the population structures of the Western countries of Europe, America, and the UK, we have an age structure such that we have a large number of old people in our communities. The very, very old, those greater than 80 to 90 years old, they're going to carry on increasing, but there will always be a small percentage of our populations. So we will have a steadily rising cost in the UK and America and Europe. The countries that are really going to suffer are countries like Brazil. 40, 50 years ago, maternal care and childcare improved significantly in Brazil. That means that there are a large number of people currently aged 60 to 40. There's a hump in the population structure, and over the next 10 years, those people will all become old enough to get Alzheimer's disease. And that is going to impact on countries like Brazil enormously. Think now about China, one of the world's most important economies. The economy that will either support you in your old age or not. In China, with vast numbers of people, they are having a rapidly aging population, and China has the unique situation of a one-child policy, which means when those people get Alzheimer's disease, there's only going to be one person in their family who's able to cope and look after them. And if that person is a wage earner, that could have very serious consequences for that person, that family, and indeed the whole Chinese, and therefore the world economy. So this is, I'm not exaggerating, one of the biggest health problems that your generation is going to face, ever. So, it started with a partnership between a doctor and his patients, different sort of partnership than we would have today, because I don't suppose that she was as active a participant in terms of engagement in research as we'd have today. But nonetheless, it's important to remember that it was a partnership, and Alzheimer wrote about this lady. Her name was Augusta Dieter, and he wrote about her in the most exquisite way. Her notes were found about a decade ago in a basement of a hospital in Germany, together with that photograph. And Alzheimer wrote the most exquisite and careful clinical history about this person, describing how she was becoming cognitively impaired, losing her memory, describing how she had hallucinations, visual and auditory, describing how she had behavioural disturbance. He described all the sets of symptoms of Alzheimer's disease that I hope you'll be encountering in your studies. Really beautiful, actually, really well described. And when you read those notes, they've been published, you can almost feel him thinking about this patient carefully, and it comes alive 100 years later. She then died, and he examined her brain, and in her brain he saw two changes, these two lesions, the plaque and the tangle. The plaque is the lesion on the left-hand side, and tangles are the lesion on the right-hand side. Do I have a pointer? Never mind. So let me use this. So this is the plaque, and it's an extracellular lesion. And that's an amyloid protein. I'll tell you a little bit more about that in a minute. Those are the dying neurites surrounding the plaque. These are neurofibrillary tangles. That's a normal neuron, normal pyramidal neuron. This is what Alzheimer described here. Can you see it? Up the side of this neuron is what he described as a flame-shaped lesion. He described it as a very peculiar substance, and indeed it is a very peculiar substance. What that substance is a protein called tau that is highly aggregated. So these are the two lesions, the plaque and the tangle, and much of the research that's been done over the past 20 years has been describing the molecular pathogenesis of the plaque and the tangle. And that research was put together in this way, the amyloid cascade hypothesis, by John Hardy when he was based in London. He then went to America and has then come back to London. And he described a set of changes that he predicted would happen, or led to many hypotheses. But the amyloid cascade hypothesis is one of the most replicated and substantiated hypotheses of how a disease forms in medicine, I would argue. It's certainly in the brain. And there's no doubt that the hypothesis, such as he described, is true. And you'll hear lots in the newspapers and the medical press about people doubting this hypothesis, but take it from me. At least in the simple sense, this cascade hypothesis is very true. What does it say? Well, it says there are genes that increase your risk of getting Alzheimer's disease. Some of those genes, the genes for early onset familial Alzheimer's disease, that's the FAD, are causative. There aren't many of those people, but they're truly causative. There are some autosomal dominant, early onset forms of Alzheimer's disease that are caused by mutations in some key genes. And if you have the mutation, you have the disease. If you have the disease and you have children, half of your children are affected, and so on and so forth, because it's autosomal dominant. Thankfully, there are very few. We did a study a while ago where we calculated there would be 100 of these individuals there or thereabouts in the UK. There are 800,000 people with dementia, of which about 100 have this rare inherited form. Then there are other genes that influence your chances of getting late onset Alzheimer's disease. I'm not going to say much about those, but they're beginning to be revealed. One of them is called ApoE, another is called clustrin, another is a complement receptor. We published, a big group published a fortnight ago, another one called TREM2, which is an inflammatory regulator. So genes increase your chances of getting Alzheimer's disease. And then, because that's what we're meant to do, I put in environment. But actually it's very, very hard to find the true environmental cause or influence even of Alzheimer's disease. Diabetes is one. So late onset type 2 diabetes increases your chances of getting Alzheimer's disease, and I'll come back to that. But there are very, very few other environmental causes other than age, which is of course dominant. And then the amyloid cascade hypothesis says that the genes in the environment alter the processing of this protein. This protein is called amyloid precursor protein. I don't want to tell you a great deal about that, other than to say it's very important. Amyloid precursor protein is very important because there's a small bit of it which gets chopped out, and that's the amyloid peptide. And the amyloid peptide is what forms plaques. And the plaques come before the tangles. And there's lots of evidence for that. We can come back to that later if you want. But for now, take it as read that the plaques come before the tangles, but it's the tangles, because they're in the neurons, that actually cause the dementia, the cognitive impairment and everything else that follows it, that Augusta Dieter suffered from. Now, a little caveat here, it might not be the plaques themselves, it might be the amyloid rather than the plaques that do the damage. And it might not be the tangles themselves, it might be something to do with how the tangles form that does the actual damage. But in general terms, you get genes and environment that influence amyloid precursor protein, which does something to amyloid, which does something to the protein that's associated with tangles, and it's that gives rise to Alzheimer's disease. Based on that hypothesis, and that hypothesis was first articulated 20 years ago, that was all you needed to know if you were a drug company to go and invent drugs. That's exactly what the pharmaceutical industry did. Based on all of the information I've just given you, was enough, essentially, to develop multiple drug development programs. They did it, and they were very successful. And people sometimes say, well, Alzheimer's disease is terribly difficult, you're not making much progress, it's much harder than for diseases like cancer. Not at all. It's exactly the same. The time from target identification to delivery of drugs to clinical trials is pretty much bang on in Alzheimer's disease as it was for cancer and heart disease and all the other diseases. There's a timeline of 10 to 20 years from when you've got the target to when you've got the drug. It's exactly the same in the disease that I work on. And drug companies, there are rather more than that, went and developed all these drugs, and when this meta-analysis, not quite the right word, when this survey was done in 2009, there were nearly 700 drugs in development. There were 50 phase 2 trials, and there were 10 phase 3 trials. Had I been giving this lecture in 2010, or the first part of 2011, I would have said, it's fantastically exciting. Thank you very much. Great. There we go. I would have said this is fantastically exciting. We've got phase 3 trials of drugs to alter the disease itself. In 5 years, I would have said, we're quite likely to have had in the clinic drugs that will alter the disease progression. Fantastically exciting. However, it's not been a good year in Alzheimer's disease research. It's not been a good year because all of the drugs that I've covered yellow have been through at least one major, massive phase 3 trial and failed. They don't work. These trials cost a lot of money. These trials are, and I'm not exaggerating here, $500 million to $1 billion per trial. Some of them, there's been more than one trial. We've been involved in some, so we've had patients that we've been recruited to that one. It's difficult to know why they failed. It might fail because the hypothesis was wrong. I don't think that's the case for reasons I won't go into. It might be they fail because the drugs don't do what we think they should do, but actually there's quite a lot of evidence, including from from patients that have gone on to die, that the drugs do exactly what we expect them to. So these kind of drugs, which are actually, for want of a better word, vaccines, they're immune clearance of the plaques. They get rid of plaques that I showed you about in mice, and when you look at people that have died, they get rid of them in the human brain. It's just, it doesn't seem to have made any difference to the patients. I think there's another reason why these trials have failed, and I think that we're doing these trials too late. We do these trials in people with Alzheimer's disease. By the time you come and see me in the clinic with Alzheimer's disease, you've lost a lot of your brain. The neurons are dead. It's difficult to say how much of your brain you've lost, but it's a significant double-figure percentage. These drugs aren't going to work for people that have lost a large part of their brain. There's lots of evidence to show that by the time you come and see me, even if you come and see me really, really early, when you first notice a problem, you've probably had the disease for 10 years or more. So we need to go back 10 years earlier than we're currently doing. For that, we need biomarkers. I've got a research group that's looking for those kind of biomarkers, and I'm not going to tell you anything about that. Instead, what I'm going to tell you about is the work that we've been doing for the past 3.5 years, trying to go a bit beyond these pharmaceutical approaches, because what the pharmaceutical companies have done is they looked at that amyloid cascade hypothesis and they said, oh, it's obvious what we need. We need a drug that stops that cascade really early on. We need a drug that targets amyloid. And that's what most of these drugs do. And actually, I'm not sure that that was the best of ideas. 23.5 years ago, when I first started doing research, I was still just finishing, really, my first registrar post, and I had the most important thing in a research career, and that's a mentor. And his name was Brian Anderson. He's since retired. I've got his job now. His retirement was nothing to do with me. And I went to Brian. I said, this seems to me to be an interesting area of science. I'd really like to work in this area. Can I come and work in your lab? And he said, well, yes, if you can get some research money, you can come and work in my lab. I said, what should I do? And he said, well, do you know what you should really do, which not that many people are doing, is try and find out why amyloid causes tangles. So he said, work in this area. Loads of people working there, loads of people working there. Work in this area, between the two. Because he said it won't be one arrow, there'll be loads of different arrows. And if you work in that area, and you're successful, you might find something useful. He then said, you're very unlikely to be successful. This is very difficult, but give it a go anyway. So 23.5 years later, I've given it a go. And a few years ago, I would have said to you, I've got the answer. It's definite, it's obvious. It's all about shaggy. Now, I don't mean this shaggy, the singer. I don't actually even mean that shaggy either. I mean that shaggy. That shaggy is glycogen synthase kinase 3, or GSK3, which in Drosophila is called shaggy. And it's called shaggy in Drosophila because Drosophilists are funny people. Is there anybody here who would call themselves a Drosophilist? Because I'm about to be rude about Drosophilists. No. All right, but don't repeat this. But Drosophilists are very funny people. They spend all their lives with this little animal, and it can be very boring because they have to do loads of breeding of this animal. And because it's so boring and they're stuck in these little spaces in not very nice labs, to amuse themselves, they give funny names to the genes that they find. It's not much of a life, but you know, you can't. And the funny name they gave to this particular gene, they discovered a mutant fly that its hairs go all the way different ways. It looks like it's having a bad hair day, this poor fly. So it's a shaggy fly. And when they found the gene that causes the hairs on the fly to go all over the place, they called it shaggy. I'll give you another instance of that. There's another signaling pathway, and it makes the hairs go right upright. So they call the mutant, the gene that causes that, they call it hedgehog. And then they found another gene that regulates hedgehog. Do you know what they call that? They call it sonic. Actually, that's quite a good joke, I think, actually. But anyway, coming back to shaggy, I did used to think it was all about shaggy, and I've spent 23 years of my life on this, and I now know that it's not as simple as that. So this was about, I suppose this was the first 5 years of research coming up with this, this experiment here. In order to try and look at that space between plaques and tangles, what I thought I would do is start at the tangle end. Remember I said to you tangles are made up of a protein called tau. I haven't got time to tell you what tau does. Suffice to say it's a very important protein in neurons, and it's very highly phosphorylated, and it regulates lots of things in neurons, including important things like how the cell body communicates with the synapse, how the cell develops. And there's many other people that work on this, including here at King's. So I was interested in tau. I was interested in how tau got phosphorylated. So I wanted to screen a model for kinases, enzymes that would add phosphate groups onto tau. So I invented a cell model. I took some cells, there are rather special cells. I put huge amounts of human tau into it, and I worked out a way that I could activate or put in lots of different kinases. And at first I was very excited because the experiments worked. I could see that the tau was functioning. I could put kinases in and I could measure the effects of those kinases. And at first I was really excited. The first year and a half was brilliant. I just loved it. But after about 18 months, I wasn't getting any positive results. Everything was negative. I could get tau into the cells, I could get kinases in, but they didn't phosphorylate tau. Even when they did it in vitro, in a test tube, they would not do it in cells. I started to get a bit dispirited. I carried on, and the first lesson you learn, if you want to be a scientist or a researcher, it's useful to be bright, but I think that's overrated. It's useful to have had a good idea, but actually I think that's a bit overrated as well. The single most important characteristic is perseverance, because at times it won't work, and the people who make it as scientists and researchers are the ones that can just carry on going. It's probably a bad personality defect, but it's one that helps if you're going to be a researcher. So I just carried on going. And purely by serendipity, a colleague of mine, who I still work with, Diane Hanger, based at the Institute of Psychiatry, on her way home, happened to go to a lecture at another institute in London. And this is another characteristic you need as a researcher, luck. She happened to go to a cancer talk by a guy called Jim Woodgett, who was working on an enzyme that he thought was important in cancer called glycogen synthase kinase 3. So she got talking to him after the talk, she took some kinase home, and she did an experiment in a test tube, and it phosphorylated tau. That wasn't that remarkable, because there have been dozens of kinases that phosphorylated tau in test tubes. But I took the kinase anyway, and without much hope, I ran it through my assay, and it phosphorylated tau. It was the first one that I'd seen phosphorylated tau, and it did so without a shadow of a doubt. This was not a qualitative difference or a small difference. This was not something I had to think about or use statistics. This was a binary result. No phosphorylation, complete phosphorylation. And I embarrassed myself actually by running around the lab. I even confess I ran up and down the stairs shouting at Eureka. It's just as well I wasn't in the bath at the time, but that would have got me into trouble with HR had I been in the bath at work. But coming back to the story, this is an illustration of this. So these are hippocampal neurons from a rat fetus, and they stained with an antibody called 88 that recognizes highly phosphorylated tau. So that's highly phosphorylated tau. And then I put on a GSK3 inhibitor, and I removed that tau phosphorylation, not completely, but nearly completely at this particular epitope. Incidentally, for those of you that do a bit of psychiatry in your training or when you're doctors, you will be given patients this drug, because this particular drug is lithium. We give it to our patients. This was given at very high dose, but actually it does it at the dose we give to patients as well, which is kind of interesting. Subsequently, other people showed the same thing in mice. So at this stage, I thought it's all about shaggy. It's obvious. And lots of people around the world have shown pretty much the same thing. Some other people showed some really exciting things, such as amyloid, or A-beta, increases GSK3 activity in neurons. And A-beta neurotoxicity, in other words, killing cells with amyloid, the GSK3 inhibitors, including lithium and a drug we also worked on that AstraZeneca produced, that protects cells. It seems to block this pathway if we inhibit GSK3. That's just one other piece of evidence making me think it's all about shaggy. So let me show you a little bit more of how we developed this in two studies. A study, first I'll show you in flies, and then a study in mice. And of course people had gone there before we did. So Jesus Avila and all his colleagues in Spain did some studies in mice where they put too much GSK3. They made transgenic mice that overexpress GSK3. These mice get learning and memory problems. They get neurodegeneration. And George Jackson and his colleagues in America, they put too much GSK3 in Drosophila, and they showed too that these animals have some neurodegeneration. So we tried to build on those kind of studies. The first thing we did was construct our own Drosophila model. So Drosophila is actually, even though I'm rude about Drosophilists, I do like Drosophila very much indeed. It's a great organism to work with for a few reasons. One of those reasons is that you can do some smart genetic tricks with it. And in this particular case, we inserted some interesting genes in a particular subset of neurons. So we can direct the gene just to neurons we're interested in. That's one good reason for working with Drosophila. And the other is it's really, really cheap. So to run a Drosophila lab, really all you need are some old-fashioned milk bottles and some bananas. The bananas don't even have to be that ripe. So you can run a great Drosophila lab with not very much. And that appealed to me hugely for all sorts of reasons. So this is the genetic trick. It's the UAS-GAL4 trap. And we express the genes we were just interested in motor neurons. And then I wanted to look at vesicles in these neurons. I'm not going to show you this data, but this is about axonal transport, how the cell body communicates with the synapse. You can see a picture of them here. We were able to label those little vesicles by green fluorescent protein, originally coming from a jellyfish, labeled neuropeptide Y. And then we put in human fetal tau, and we put in a mutated Drosophila shaggy. And it's just as well this sort of stuff doesn't get to the Daily Mail undigested, because I've actually made a Frankenstein mutant that is part fly, part jellyfish, part human fetus, and part mutation shaggy. And that doesn't read so well when it comes back to you from the journalist. But that's the model that we did, and it affects the flies quite a lot. Let me show you first if this works. first in larvae. So what you're now going to see is a normal Drosophila larvae, hopefully, walking or crawling down a little path we've made for it. You can see some peristaltic waves coming down its body, and you see how its tail flicks up. So that's a normal Drosophila larvae. And then in the next experiment, here we've put in tau. So we're now modelling some aspect of Alzheimer's disease. It's not really Alzheimer's disease. The thing doesn't lose its memory and get lost. But it does have a very particular phenotype. So you can see it's much, much slower. It's lost that typical tail flick, and the peristaltic muscular waves don't seem to somehow go all down the body. So that's the larval phenotype. We then looked in the adult animal. Again, flies that have got too much tau, transgenic flies. This is geotaxics. So we've got the flies in this test tube here, and we connect that test tube to that test tube. Then ever so carefully, because we wouldn't want to hurt the flies, we whack them down to the bottom. So all the flies are falling down here. I was joking, by the way, when I said we don't want to hurt flies. We kill them. And then they crawl up the test tube, and we count how many flies get to the top test tube in a certain amount of time. And that's geotaxics. The flies tend to crawl up there. And I'm sorry if I sound callous about how we treat the flies. We do it for the best possible reasons, of course. And in fact, this project did have lay people from the society with us, just to check that everything that we were doing was all okay. And they were happy with how we were treating the flies. Honestly, I kid you not. So this is the results of the adult experiment. And if we look at the pink line first, this is the geotaxis in normal flies. And every time I look at this, I think that flies are very, very like people in lots of ways. I'm not meaning for a moment that my colleagues are like fruit fly maggots, or there are one or two. But I actually mean, if you look at how they start off, this is the equivalent of a fly toddler. And for any of you that have had any contact with toddlers, you're running around all over the place, this is a very, very active fly. This is the equivalent of a fruit fly teenager. I have some experience of human teenagers, and they become, and I realise that many of you will, because I'm on dangerous ground here, and I realise many of you are not that far away from being teenagers. But in my experience, teenagers are fairly slothful things. They spend all their time in their room, they're on Facebook, they're certainly not doing their homework, and they don't pick their dirty clothes off the floor. Exactly like a fruit fly larvae, which is not moving around very much. The fly then gets more and more active until it reaches the pinnacle of its life. The pinnacle of its life is when it's most intelligent, most thoughtful, kindest and active. And if you want to know the human equivalent age, it's 52 and a half. And then they get worse, the flies gradually deteriorate, and this is old age in a fly until they're not moving around very much. So that's normal fly phenotype for geotaxis. This is the fly that we've disrupted its tau. We've put in transgenic tau, so it's overexpressing tau. So we're trying to model the dementia. And you can see the first half of its life, it's exactly the same as the wild-type fly. In the second half of its life, it reaches the floor of this particular test. We can't really measure change after here. So there's a very distinct late-life phenotype in this animal that we can then use to do our other experiments. And the other experiments that we want to do, of course, to prove the point that it's all about shaggy, is to treat it with the drugs. So if you look at the black line first, this is the control. This is what happens when you put in too much tau. This is what happens when you put in tau plus shaggy. You can see the phenotype gets distinctly worse. So that's very good evidence that it's all about shaggy. We then treat it with GSK3 inhibitors, and we reverse that phenotype. And this is really important. We reverse the phenotype even when we haven't put in GSK. So the endogenous, the normal fly GSK3 is responsible for the phenotype in that transgenic tau animal. We can restore those flies, we can cure them. And that was really all that we needed to then progress clinical trials, which I'll come back to right at the end. But what this experiment showed, for the first time I think in vivo, was that we had a GSK3 dependent tau-induced phenotype without tau aggregation. So I was really, really happy when we published this. It was clearly all about shaggy. So then I became even more bold, if you like, and we asked the question then, well, if shaggy or GSK3 is responsible for Alzheimer's disease, is it responsible for memory itself? And there were actually some good reasons to think it might be. So we collaborated with the people that had produced this transgenic mouse in Spain. And this mouse has increased tau phosphorylation, has deficits in learning and memory, and it has neurodegeneration. But it has all of those when it's about 9 to 12 months. So what we did was we took those mice really early, at about 3 months, 4 months, thereabouts. So before they had any neurodegeneration, and we asked, is the memory normal in the mice before they have neurodegeneration, or is it abnormal? And actually we wanted to go beyond memory. There is something called long-term potentiation, which is the nearest equivalent we've got to a cellular or molecular model of what we think is happening in cognition. So I need to explain to you long-term potentiation. It's a fantastic thing. It was discovered by Tim Bliss at the National Institute of Medical Research in 1973, it was published. And it's still the best understanding we've got of how you think. So essentially, if you take a neuron and you give it an electric shock, you know that you can measure an output, right? So if you give it a shock, measure an output, and you give it another shock, you get the same output. If you give it a third shock, you get the same output. And you can carry on doing that for as long as you like. Give it an electric shock, you'll get an output. But if you now give it a tetanic shock, so instead of giving it one electric shock, you give it a rapid burst, so a really high input, and then measure the output goes up. And then the clever thing, if you then give it another single shot, the output is still high. And it stays high for hours and gradually fades. So it's as though that neuron has remembered getting a high input. That's called long-term potentiation. You've potentiated the output, and it's long-term, meaning some hours. And that's been measured in single neurons. It's been measured in every organism that you can do these kind of studies. Worm-type organisms, flies, and as I'll show you, even in mice. It's hard to do in higher organisms after that. So we asked the question, is GSK3, shaggy, responsible for long-term potentiation? So this is the experiment. This is a control mouse, and this is what long-term potentiation looks like. Little shock, little output. Little shock, little output. Little shock, little output. Big shock, big output. Little shock, big output. Little shock, big output, and so it gradually fades away. 2 hours later, you can still see some memory, but not much, and it carries on for a little bit longer. So that's normal mice. And that's the GSK3 mice. You can see that it still has a little bit of long-term potentiation, but it's pretty much lost by about half an hour. So we've not abolished, but we've severely attenuated or decreased long-term potentiation by overexpressing GSK3. You'll now want to know what happens if you give a GSK3 inhibitor. Well, amazingly, if you give a GSK3 inhibitor, you restore, that's the green line now, you restore those mice to complete normality. And at the same time as we were doing this, another group, led by Graham Collingridge in Bristol, that are world experts, he did his PhD with Tim Bliss, and they published not the same thing, but are very related things. So our two groups came up with the same finding simultaneously and not knowing that we were both had the same idea and we were working on it together, which is another lesson that happens a lot in research. If you have a good idea, chances are somebody else has had that same idea at that same time. So here, by this point, I'm really ecstatic, because by this point, it's so obviously all about Shaggy. Shaggy has to be pretty much all you need to know about Alzheimer's disease, and in fact, memory. And then it got a bit more complicated. So at this point, as I say, we're very excited. We think that GSK3 is a tau kinase. I didn't tell you this, but other people think it's an APP kinase. We show that it's induced by amyloid, it mediates neurotoxicity, etc., etc. So we go on to do some thinking about clinical trials. This is from King's. We did a 1-year clinical trial. We gave lithium to people. Lithium's quite a poisonous, dangerous drug to give people. You have to be very careful to measure blood levels. People can go into the toxic range really quite quickly. And if you become toxic, the window is relatively narrow. If you become toxic, you can die, and people die of lithium from time to time. If you treat within that window, it's very safe within that window. The question is, you're treating old people with memory problems with a drug where you have to get within a window, otherwise it could be fatal. Could we do it? Well, we showed we could do it. Harold Hampel did a 10-week trial and showed no change in biomarkers. But Orestes Valenza in Brazil, used to be a research fellow with me, did a 1-year trial which he published last year and showed some benefits in people with mild cognitive impairment. In a randomized controlled trial, these people had decreased phosphorylated tau in their spinal fluid, and they had some improvement in cognition. So, you know, by now, I think that I could almost retire at this point. Everything seems to be about Shaggy. So we went on to ask a question, well, can we do what I was originally told would be a good idea, to work out how amyloid increases tau phosphorylation? Could I work back up the pathway? So we became interested in how Shaggy, or GSK3, is regulated. It's regulated by two routes, by Wnt, or wingless signaling, I'll come back to that, and by insulin signaling. And you remember I told you at the beginning, diabetes is one of the very few environmental risk factors for Alzheimer's disease. So diabetes increases your risk of Alzheimer's disease, and there are a couple of big studies going on in America at the moment of giving people nasal insulin as a potential therapeutic for Alzheimer's disease. So we decided to model that, again in mice, and this was work that I did with Dominic Withers. We took a mouse that has the IRS2 gene knocked out. So IRS2 is part of the receptor complex for insulin, and these mice, the homozygous IRS2 double knockouts, have diabetes. I mean, it's not quite diabetes, of course, but it's a close rodent model of human type 2 diabetes. So we took those mice and we crossed them with a mouse, the TG2576 mouse, that has plaques. The hypothesis was, if those animals have lots of amyloid in their brain and they don't have a good insulin signaling system, would they then get full-blown Alzheimer's disease? This is a bit complicated, but to make it simple, just look at any one of these really, but let's look at this set of 4 results. And what you have here is a measure of tau phosphorylation, and this is in normal mice, very little tau phosphorylation, if any. This is the Alzheimer model mouse, and this is the problem with Alzheimer's disease models in mice. They have lots of plaques, but as you can see, they don't really have much, if any, tau phosphorylation. This is the diabetic mouse. It has tau phosphorylation, quite a lot, significantly more than a normal mouse, which is what we predict. This is the cross. So this is the mouse that has both plaques and diabetes, and it has a massive amount of tau phosphorylation. So that seemed to be good evidence that regulation of GSK3 or Shaggy was important. And maybe this explains why people with diabetes get Alzheimer's disease. This is where it all starts to go wrong. I mean, it goes wrong in a good way, because of course life is more complicated than that. But we had quite a difficult 5 years in the lab as we uncovered something that wasn't in line with expectations. And that's my last message, as it were, to your students. If when you're doing research you find something that is not what you expected, your first reaction will be despair. Actually, what turns you into a good researcher is taking the unexpected and making something from it. Because when you find something that challenges your own ideas or firmly held beliefs, that is your most valuable moment in science. So we thought, well, it's obvious, isn't it? Insulin is part of this system because it regulates GSK3. So the regulation of GSK3 through Wnt should be part of this as well. And another group published that Wnt3A blocks amyloid toxicity, all in line with what we expect. It's all perfect and everything was fine. Except Wnt signaling is not simple. So I don't expect you to read all that, but that's just to illustrate that it is actually really, really complicated. And Wnt signaling doesn't go down a single route, it goes down multiple different routes. And to cut a long story short, they have different effects. I mean, I'm going to explain this quite quickly, and I don't expect you to follow all of this because this is a bit complicated. If you wanted to read it, it's got published in the current issue of Molecular Psychiatry. And it's the result of maybe the last 5 or 8 years of work in my lab. So another group published that this interestingly named gene, DICOF1, which is an inhibitor of the Wnt pathway, was increased in Alzheimer's disease. So we thought, great, yet more evidence that it's all about shaggy. Let's explore that. So we think then GSK3 lies here. I'm interested in, can we understand all of those steps? So here's the experiment. First of all, we showed that what this group had just published was right. Sure enough, when you add amyloid to cells, they increase DKK1, which should be inhibitory on GSK3 activity. So we did a whole genome study. What we did was we took some cells, some neurons, and we applied amyloid to them and measured every gene change in those cells. We then took some DKK1 or DICOF1 and did the same thing, applied DICOF1 to the cells and measured every gene change. We then put those 2 gene lists on top of each other. And we said, what are the genes that are the same? Because if you have a gene change that's induced by amyloid and one that's induced by DKK1, that should be the absolute pathway to Alzheimer's disease. Of course, I knew what the answer would be already. It would be GSK3, right? It was obvious. But we'd do the experiment anyway. So we did the experiment, and we got a massively increased overlap, increased above chance. So large an increase is that in the paper, if you read it, it says p less than 0.00. And the referee said, well, 0.0 what? What's the actual p-value? I said, we don't know. It's such a rare event. The statistical test that we use doesn't go small enough. It's p 0.0 recurring, as far as we can tell, forevermore, because you just don't get that level of significance. Actually, we didn't need statisticians. We didn't need bioinformatics to look at these 30,000 genes, because you could just eyeball it. If you look at the top 10 genes, you can see in the amyloid list and in the DKK list, a load of them are the same. So EGR1, NAB2, cyclin D1, CFOS and KLF10, they're the same in both lists. The p-value for that is 10 to the minus something or other. It just doesn't happen unless you really have found something. So we thought we'd really found something, but it wasn't GSK3, which was very alarming to me. We then went on to prove it, so we then took some cells, we treated them with amyloid and they died. And if we knock down the genes that we found, the cells survive. So this really is a pathway. We then went on to find out what other genes are in the pathway, and it's not GSK3, we think it's these two genes, this family of genes, junk, and that tells us that it's not canonical Wnt signaling, it's planar cell polarity signaling. The details don't matter. Every new bit of evidence tells us it's not as simple as GSK3 or shaggy. So then we went and had a look to see if we could see our genes in a mouse with amyloid plaques. Sure enough, we can see the genes elevated in the mouse model. But when we look at a different model of neurodegeneration, one further downstream, the tau mouse, we can't see those gene changes. So we then went and had a look at patients' brains. We found the same thing in man as we found in the mouse. The genes that we're interested in are up in amyloid diseases like Alzheimer's disease, but they're not altered, with one possible exception, in frontotemporal dementia. So we then made another mouse. We then make a mouse that is overexpressing DKK1. So this is the mouse here, it's got DKK1 gene and protein, you can see that there. And this mouse has high levels of tau phosphorylation and it has memory impairment. So we think that DKK1 and some of these other genes are actually the pathway, and it's not as simple as Alzheimer's disease. So that's just to show you the tau phosphorylation going up and the memory impairment in these mice. So it turns out that I started 23 years ago looking for a tau kinase. I think that tau kinase is in fact GSK3, and everything that we did after that suggested that the GSK3 or shaggy is the key point in the amyloid cascade. Work we've done over the past 5 years, somewhat painfully but very interestingly, has told us that although the GSK3 led us to this, it's not as simple as that. So what we actually think is happening is that A-beta or amyloid is setting off in train two pathways. One that goes via P53, DKK1, and all of these interesting proteins, and another that goes via GSK3 to tau phosphorylation. And today, right now, I just don't know whether that tau phosphorylation is contributing to the neurotoxicity or not. I do know that this pathway is contributing to neurotoxicity. So if you wanted a drug for Alzheimer's disease, I would say look on that part of the pathway rather than that part of the pathway. And of course that's exactly what we're doing. So the work that I initiated 23 years ago, and Diane Hanger initiated at that time, suggesting that GSK3 has led on a translational pathway to drugs that are currently in phase 2 trial. I know the results of those trials. We led the European phase 2 trial. I can't say what the results are, but I think actually they're more in line with the work that we've been doing over the past 5 years. We are about to start with colleagues in South Africa a trial of lithium in HIV-associated dementia. There were reasons to think that it might not be a bad idea to do that. There are others that are looking at diabetic drugs and insulin therapy in Alzheimer's disease. This in phase 3 trial failed. I hope that doesn't fail, but we'll see. Actually, I think that inadvertently we set off on a translational pathway that was flawed. I think that we now know what the pathway is. I understand how we got to that point, but you won't be surprised to know that we are initiating, we're some way through actually, a new novel drug development program, and I'm now not concentrating on shaggy, but on the DKK1 pathway. So it's been an interesting translational journey for me. I hope that you found, at least if not the details, some of the way that we moved from one bit to another of interest. And of course the real pleasure for me has been working with a great number of fantastic scientists over the years. I'm closest associate Richard Killick, but many others that I've worked with at King's and elsewhere in London and further afield. Thank you for your attention.
problem is that you're not targeting the process too late, that it targets patients who, like, they already have too much neurodegeneration to go backwards. So what about targeting, like, the earlier bit of the pathway where you can, I mean, if possible, to pick up the people early on?
Yeah, no, that's exactly what I said, and that's exactly what I think. And in the other part of my lab, we're trying to find biomarkers, blood-based biomarkers, CSF markers, and neuroimaging markers. And the real reason why we're doing that is not for diagnostics, because as doctors we're pretty good at diagnosing Alzheimer's disease. We need a biomarker that will pick people up when they have early pathological changes in their brain that have no clinical symptoms, so no doctor can pick them up and do the trials in people really early. To do that, you need the biomarker because you couldn't do it in an entire population, but it also is a substantial logistics and cost problem. Because it means we have to think about doing trials of 5, perhaps even 10 years long, and if a trial costs $500 million for a 1-year trial, then do the math. It's really worrying at the moment, because if that's the only way forward, governments and pharmaceutical industry are all asking, can we actually afford to do clinical trials in Alzheimer's disease? But that is exactly the only way I think we will make progress, is by doing studies much earlier in the disease course.
I'll also use Alzheimer's, so I've got another question now. Some of my patients have been in the Whitehall study. I understand that, hang on, what's going to be about 50 years, but the patients who declined and developed Alzheimer's showed earlier memory loss than those who didn't. So is there something about having a more sophisticated memory test that is on that might identify something cheaper from Alzheimer's?
Yeah, so it's a good question. The question is, work in the Whitehall study, which is a cohort study of civil servants that was looked at in 1946, I think, for the first time. When you go back retrospectively, the people who subsequently end up getting Alzheimer's disease had some memory change even before they had dementia. So the question is, could we have a better cognitive test, a more sophisticated cognitive test than the very simple ones we do currently use that would pick these people up? The answer is some people think that we might. I suppose the leading group that's doing that is Barbara Sahakian and Trevor Robbins in Cambridge. Actually, Barbara did her original work here at King's many years ago, and they have a computerized test called the Cantab battery, and she and others think that that would be a very good way to pick these signs up earlier. I think the jury's out, to be honest. It might help bring the diagnostic process early a bit. Whether it brings it forward a decade or so that I think we need to do it, I think it just requires further testing. And there's a very big study that's just been funded by the TSB Catalyst that was in the newspapers about a fortnight ago. The Daily Mail had it on its front page, that is going to be testing that kind of thing, and King's is part of that multi-sense study. I'm not convinced it's much cheaper, that's the only thing I'd say, because the thing about cognitive tests is they do require people. They require psychologists, and a psychologist is a lot more expensive than a blood test, however you pass it. But I don't think cost is the limiting factor here. The limiting factor is, can we invent a test that's good enough?
If well-validated biomarkers do get developed, would you go back to GSK3, or have you moved on to the more signaling pathways in terms of...
Well, you've read me right, of course. I can't leave it alone, and I'm not saying it's unimportant. I do think it's involved in tau phosphorylation. So yes, of course, we would try and go back to it. There are other reasons for being cautious about GSK3. It does a lot of things. It's involved in lots of signaling pathways. One would predict a really effective GSK3 inhibitor might actually be quite toxic. Nonetheless, I think it's still in there as a possible therapeutic. I just think we can improve upon it as a target.
Sorry.
Have you been in any studies in patients who've been prescribed lithium to see whether people who've had bipolar depression or whatever they claim to prescribe for, whether they can actually prevent Alzheimer's?
Yeah, it's a really smart question. Yes, they have. I know of 3. So one, in the UK, tried to use the GP database. And it was negative. It didn't show any protective effect of lithium. And although it looked at a very, very large number of the population, like tens of thousands of people, lithium is prescribed so rarely that if you look at an entire population, there are very, very few cases of lithium prescription. But it was negative. The alternative way of doing it is a matched case control type retrospective study, and I'm aware of 2, one from China, one from Brazil, and they both looked at people, as you suggest, with bipolar disorder, and there are multiple different drugs you can use for bipolar disorder. The 2 commonest are sodium valproate and lithium. So they simply looked at people that have had either lithium or sodium valproate, and actually the incidence of Alzheimer's disease was higher in the people who had sodium valproate than in lithium, pretty much as you're implying with your question. Problem with it is it's observational and it's retrospective. And the only way to test those kind of hypotheses is with a fully blinded, randomized controlled trial. But it was a good idea.
You mentioned certainly looking down the shaggy line of research. That was focusing on the tau phosphorylation and the tangle formation as the neurotoxic destructive phase of the disease. But I remember you saying something about actually the neurotoxicity of the degeneration may be caused by the plaque formation. What kind of research has there been to date looking at that part of the process?
Okay, so if I go back to here, what is, well, it's not entirely clear, but almost certain is that amyloid itself is neurotoxic. It's actually quite a difficult protein to research because it aggregates, self-aggregates in a test tube. And so, you know, when you're doing these kind of studies, you want homogeneity and reproducibility. It's very difficult to get that with amyloid. That aside, almost everybody agrees that amyloid is toxic. The question is, how is it toxic? So, I think that what we're showing is that amyloid is toxic through these pathways, and I think it is true. So I didn't put amyloid, but I sort of am assuming amyloid is up here. And what we've shown is that amyloid does kick this pathway off. And I do think that GSK3 is responsible for amyloid-induced tau phosphorylation. I'm just not clear in my own mind any longer, and there isn't very much evidence actually, that this is responsible for the toxicity. And I think we were led to this pathway because we were going up this direction. And the assumption that I had was that this would be straightforward tau phosphorylation. And that just seems, you know, I just think that's probably not true actually. So I think amyloid definitely is neurotoxic, and our best bet is it's neurotoxic through that pathway.
Is there any connection with diseases where you see either only amyloid collection or only tau phosphorylation? Because I think Parkinson's was where Parkinson's is one where you have only tau and no amyloid. I mean, I'm not sure about this, but is there any correlation with such?
Yeah, that's a much disputed question actually. So there are some patients, they're not very common I have to say, but there are some patients reported in the literature who have large amounts of amyloid in their brain and no dementia and no tau phosphorylation. That's really interesting, and nobody can explain that, but it is an observation. So what is clear is that the amyloid is causing neurotoxicity, but not in everybody. In 99.9% of people, but why are those people protected? That's a really interesting question. There are some disorders with tau-only pathology. Actually, Parkinson's is not one of them because it has synuclein pathology, but there are some disorders like frontotemporal dementia, and actually non-dementia disorders like progressive supranuclear palsy that have the tau pathology only. And in terms of our pathway, we think that, you know, there are so we think that the pathway that we're exploring is here, But if you get a mutation in the tau gene or some other causes, you can enter this disease pathway here. So you could have an arrow coming down here from a tau mutation, which gives you tangles and gives you dementia, but it doesn't involve any of the amyloid pathology. And in fact, the animal and human studies that we've shown show that you don't see the set of genes that we're inducing with amyloid in those mice or individuals.
Okay, well, you can see we are in this research quite